I'm a professor in mathematical and statistical ecology at the University of Jyväskylä in Finland. And as you see from the program here, I will start by first giving a very brief introduction to what is Lifeplan. Now, if you will have any questions or comments during these presentations, please place them into the chat. So we will do our very best to reply to them in the chat already during these presentations. And then in the end, we will have a questions and answers session where we can address at least some of the questions in more detail. So what is Lifeplan? Lifeplan is an ERC synergy project that we have been running for the last 6 years. The funding period will actually finish now in the end of this month, which motivated us to organize this webinar and tell about the results that we have obtained. And as I'm sure many of you know, ERC synergy projects are meant for a group of 2 to 4 PIs working together and bringing different skills and resources to tackle ambitious research problems. Now, in our case, we have 4 PIs, including David Dunson, Tomas Roslin, and Brian Fisher, who all are present here today, except that David is substituted by Jennifer, who will give that part of the presentation. Now in Lifeplan, we have combined the development of big data statistics with biodiversity research. And what has especially motivated us are the methodological questions of how to best obtain systematic biodiversity data and how to best use such data for inference and prediction. Now the Lifeplan project has been implemented by this really fantastic group of researchers, including PhD students, postdocs, and also some more senior researchers. We have recruited this research team to cover this multidisciplinary expertise that has been needed to successfully implement Lifeplan, and especially to reach this close interplay between statistics and biodiversity research. Now, this is my one-slide summary of what Lifeplan is all about. So through collaborative effort, including several hundreds of researchers, we have implemented globally distributed sampling with DNA, image, and audio-based methods. And this has resulted in a really vast amount of data. So in our understanding, the Lifeplan database is currently maybe the largest systematically collected biodiversity database, where exactly the same sampling methods have been applied in all of these locations on a weekly basis over the past years. Now, the first step in the data analysis is to identify the birds and bats from the audio, the mammals from the image, and the fungi and the insects from the DNA barcodes. This is highly challenging, not only because of the large amount of the data, but also, for example, because most fungi and insects are still unknown to science. And then, simply put, the second step is to apply statistical approaches to the data with the aim of improving our understanding of the processes that create spatiotemporal variation in biodiversity. Now, after this very brief introduction, I think we are ready to discuss the Lifeplan project in more detail. And next, Bess Hardwick, who is one of our really fantastic coordinators, will tell about the team effort of globally distributed sampling.
Thank you, Otso. Share. You see my slide? Yes. So hi, yes, I'm Bess Hardwick, and indeed I'm one of the coordinators. I'm based here at the University of Helsinki. I'm going to tell you first about a bit in more detail about the Lifeplan sampling design and how it's designed to address different ecological questions. After that, I'll talk about standardization. Standardization is a major part of Lifeplan, and that's how we make the data from all over the world and from different environments comparable. So you can actually do stuff with it on a global scale. Then I'll talk a bit about the spatial coverage of Lifeplan, including how that affects then the temporal coverage of the Lifeplan data. And at the end, I'll mention some important things about the ownership of the data. The Lifeplan sampling design, first of all, is on a bunch of nested scales. So we have first the global scale, where we have sites all over the world. And then we have these smaller nested scales. So in the Nordic countries and in Madagascar, we have what we call the national scale, where we're on about 1,000 kilometers. And then in one corner of Madagascar and one corner of Sweden, we have these decreasing scales going down to 0.5 kilometer, where the sites get closer and closer together. And the purpose of this is that we wanted to be able to look at what actually are the relevant spatial scales for various ecological phenomena. A second major element is that we have slightly different kinds of sites. On this map from my recently published paper, we have different kinds of sites in different colors. So we have green circles and black squares, and these are natural and urban locations, we call them. So on the global scale, the teams were asked to find a natural location and an urban location that are otherwise comparable, same kind of climate and other conditions, about 20 to 50 kilometers apart, and they alternate yearly between the natural and the urban location. And then those purple dots on the map, those are permanent. So in Madagascar and the Nordic countries and a few other places where there really aren't any urban places in existence, you have these permanent sites where the equipment doesn't move and sampling is done continuously in the same place. And this natural versus urban comparison enables us to look at the effects of human influence and human footprint on biodiversity. At every Lifeplan site, we use 5 different sampling methods. We have cyclone samplers that collect fungal spores from the air, and we take soil samples to see the fungi in the soil. Malaise traps passively collect flying arthropods. And then we have image and audio from camera traps and audio moth audio recorders. So we are focusing largely on arthropods and fungi as very large and poorly understood groups. And with image and audio recording, we get the sort of larger, more charismatic megafauna, mammals, birds, mostly. And this also does qualify as long-term data. The sampling ran for 5 years, from 2021 to 2025. So you can also start seeing more long-term patterns. And it mostly was conducted year-round. So next I'm going to talk about standardization, what we did to standardize the data. This starts with protocols that we developed together with the teams at the very start of the project. We had some meetings to refine the protocols to find field sampling methods that would work everywhere. And our protocols are published. We published the full Lifeplan setup, which describes in more detail what I just talked about on the last slide in a +1 paper. And through that +1 paper, you get links also through to these individual field guides for the 5 different sampling methods. We've also published our DNA sequencing protocols. So our Malay sequencing protocol is published. The cyclone sequencing protocol was published before Lifeplan already. We're following the same protocol. And we will be publishing the soil sequencing protocol as part of a soil data paper, I think. So Lifeplan it's not just a data set, but it's a method. And we very much hope that people will take up the method post Lifeplan, as the project is now ending, and use it to sample biodiversity in a way that will be comparable with this existing very large data set. A core of the standardization is to use the same sampling equipment and the same sampling methods in all these different environments. So we provided the sampling equipment to teams around the world. We shipped it to them. Here you can see malaise traps in various different environments dealing with moisture and snow and sun and heat and all that. We also had the same sampling frequency in adult plots. So the basic rhythm is weekly. We take cyclone samples weekly, actually 2 samples a week usually, but we used 1 for now. Also, the malaise trap was emptied weekly. Camera traps and audio recorders also visited weekly, and soil samples taken 4 times a year. The plot design was also the same everywhere. We started with this 100-meter square plot with traps at all 4 corners and in the middle, with just 1 cyclone sampler and malaise trap in the middle, but then everything else at all 4 corners and the middle. We did have to adjust the plot design in the 2nd year because we were breaking down so much equipment and collecting so much data. We cut 2 corners and we ended up with this transect with 70 meters roughly between the points. But this is the plot design that was then followed at all Lifeplan sites. For standardization, metadata collection is extremely important. What we did is that we had our own app, the Lifeplan mobile app. And every time a team visits a site to place a bottle in a trap or collect it or place a memory card or collect it, they scan a QR code on the trap and on the sample container or memory card. This created an activity, and from the phone, it takes date and time and the location of the phone. And this is all collected by the app into a central database. And that's how you get the central metadata that you always need with all biodiversity data, which is time and place. And then as the data itself came in terms of DNA sequence data and images and audio, it was connected to the metadata. I'm not going to talk much about this because others will talk about it, but I will just mention that an important part of standardization is that the species identifications are done in an automated way through machine learning methods so that we don't get local biases of varying taxonomic expertise. Okay, then I'm going to talk about our spatial coverage. The way we found sites is that we put out a call for teams anywhere in the world who would like to join. And just to point out that what we were asking them to do, we were asking them to commit to 5 years of visiting a site at least once a week, but most of them visited the site 3 times a week, year round, and carrying car batteries and uploading data and charging batteries and building cages and driving up mountains and everything else that they had to do. And they also had to find a natural and urban location that would both be accessible enough for them to do this. And we provided them with the equipment and carried the sequencing costs, but otherwise they were doing this on their own dime. And considering this, it's amazing the global spatial coverage that we did manage to get. Teams around the world put in a huge amount of time and effort in all kinds of conditions, weather conditions and remoteness, and solving every possible problem in practice. And these were also one of the most challenging areas that we sampled in was Madagascar, where the sort of temperature and moisture conditions are very challenging for equipment. Also, infrastructure and road conditions are very challenging, and there we had our own sampling teams. but there they had to do a lot of work in terms of security and protecting the equipment. And I always talk about these practical challenges with sampling because it does affect then the temporal coverage. These local conditions affect that very much. So this is, in this picture, we have temporal coverage of malaise sampling throughout the Lifeplan project. So on the y-axis, we have all the Lifeplan sites ordered here by latitude. So north is up and south is down. And on the x-axis, we have time. And you see that it separates into these 5 blocks that are the 5 years of the project. We've got 2021, 2, 3, 4, 5 on the x-axis. And each of these little squares is 1 week at one site. And a couple of things you notice straight away is that it is gappy. You see a lot of little holes there between the squares. And another thing that you might notice is that the length of the sampling season changes. If you go right to the top, you see those little triangles where the summer gets shorter and shorter as you go further up north. So things like this, like weather conditions, equipment breakage in the tropics, cameras break down at an alarming rate. Some sites are very remote. They might only have access part of the year. They might be covered in 2 meters of snow for several months. And of course, as we live in the real world, there is always the reality of bureaucracy and shipping, which caused some sites to be able to start only later than others. And this is just to say that the Lifeplan data does have gaps. It has different lengths of sampling season, and in some cases, the different numbers of years. Not all sites were able to sample in all years. And this is caused by huge differences in the local conditions. So after all that work that the sites put in, including getting all kinds of local permits, all that maintenance, all that setup, all that work, of course, they do own their own data, and that's very important to keep in mind. We co-author publications with the Lifeplan teams, and they get to decide what we can do with the data. So we have permission to do the things that we agree to do within Lifeplan, but anything additional, the global teams decide for themselves. We are going to be publishing Lifeplan data as open access data papers, together co-authored with the team. So at that point, it will be available for anyone to use, but the teams get a little head start to start working with their own data. The Lifeplan project, as ourselves, we control and own only the data from Madagascar and that little corner of Sweden near Uppsala. And that's it from me. And I think next we have, is it Jennifer next?
Can you see my slides? So
Yes.
I'm going to be presenting on behalf of the Duke Statistics arm of the Lifeplan project, led by David Dunson. And practically, I'm going to be giving a high-level overview of some of the statistical methods development that's come out of this project. But really, this is a story about modern ecological data pushing statistics forward. We're increasingly encountering high-dimensional data, sparse data, and data that's structured in really complex ways. And this has posed a lot of challenges and opportunities. So throughout the Lifeplan project, a key focus has been on improving statistical inference for high-dimensional, complex, and noisy data. And of course, these methods were initially motivated by Lifeplan. The data collected here is standardized in a lot of ways that makes it really favorable to statistical analysis, but still methods are critical to deal with issues of measurement error, just the sheer size of the data, the dimensionality, and the sparsity that is natural when you're doing biodiversity monitoring in a changing landscape that's dominated in many ways by rare species. But these aren't just ecological problems. These are issues that arise in many other domains across the sciences, engineering, technology, and finance even. And through the course of the project, we've developed a rich toolbox of statistical methods to transform inference from modern ecological monitoring data, and have also built a really strong theoretical foundation and improved the broader statistical inference toolbox. So today we're going to walk through a few illustrative examples, the first is to do with scaling up JSDMs. And the idea here is just to make these JSDMs work for, I'm sorry, go back, massive data sets with hundreds of thousands to millions of species. The next project involves interpretable AI and deep latent structure modeling. We're going to zoom out and look at some principled biodiversity inference, so inference on biodiversity measures globally. We'll look at some fundamental developments and modeling how networks vary over time and space, and fundamental developments in factor modeling, allowing concurrent ordination and simultaneous or co-clustering of species and sites. So starting with scaling up JSDMs, the real need here has to do with the scale. We need methods for analyzing massive biodiversity data sets with millions or hundreds of thousands of species. And a key feature of these data sets has to do with the dominance of extremely rare species and species discovery as sampling proceeds. Typical approaches to joint Bayesian modeling of species become intractable at this scale, and they also don't accommodate new species discovery. So a lot of the methods that have been developed in Lifeplan have been motivated by those two things. And one motivating data set we can look at is the Malagasy arthropods. So we have malaise trap data from 53 locations in Madagascar with almost 3,000 samples and 250,000 different species, many of them very rare. The first method that we're going to be looking at here hide my meeting controls, sorry, there we go, hopefully that's better. Takes a note from some recent success in transfer learning to basically borrow information from common species to improve inference about rare species. And the idea here is really to build a backbone model based on common species or species that are repeatedly observed, and then to transfer information from that model to a full model involving more rare species. So first extracting latent factors for the habitat and learning the parameters for the more rare species, borrowing information from the common species on the basis of phylogenetic or trait similarity. So the result of all of this is that we're able to massively scale up JSDM-like modeling to millions or certainly hundreds of thousands of species. Let's see. Advance, there we go. Another approach in this thread of speeding up JSDMs comes from factor pre-training. So this is based on work led by Lorenzo Mauri. And the idea is to obtain point estimates for model parameters, so regression coefficients, factor loadings, and latent factors, and to also provide uncertainty quantification for those parameters of interest conditional on the estimated latent factors. This method uses a mix of optimization and Gaussian approximation to avoid MCMC, and also provides a method for variance inflation so we can make sure that the uncertainty quantification is well calibrated. So in addition to providing excellent performance for the Lifeplan data, we've developed really strong theoretical support for this. So it provides a more general template for scalable inference in a broad Bayesian latent factor modeling context. Sticking with the theme of abundance modeling is this work on deep latent class regression. The idea here is that latent class regression can be a really powerful tool for categorical data. We can think of binary abundance data as an example of that. But when you get to the scale that we see with the Lifeplan data, traditional methods become really unreliable. So this method, led by Yuan Zhao, takes environmental covariates and multiple different layers of binary latent attributes, and results in much more reliable inference for high dimensional Y. And something that's really useful that comes out of this is that the attribute, the latent class allocation, induces a clustering on sampling that's dependent on covariates. And it can allow us to do inference on things like regions of common profile and produces much more geographically and ecologically coherent regions than we see in more simple traditional latent class regression like you would have on the left. We see much more coherent regions on the right. And then zooming out from abundance modeling to looking at how biodiversity varies worldwide, we have Hubble regression, which provides a principled tool for covariant dependent modeling of alpha diversity. The key idea here is the observation that the distribution of species richness under a Dirichlet process follows an exponential family that depends on alpha. So this provides a really elegant link between ecological theory and statistical modeling. And this work, led by Ale Zito, allows us to answer long-running questions about the relationship between environmental covariates, evapotranspiration, or the human footprint on biodiversity worldwide. Another problem the team has worked on is network modeling. So here we're not just interested in where the species are, how abundant they are, but how they're interacting. And in particular, we're interested in how networks, interaction networks, vary over time and space. So we're imagining here that we have a collection of different networks observed throughout space and time. Standard latent space network models require estimation of a huge latent node attribute tensor across time or space. This becomes quickly computationally unfeasible. And additionally, when the data are really sparse, estimation just becomes unreliable with such a high parameter space, high dimensional. Our approach has been to instead take a low rank tensor decomposition of this latent attribute tensor. So we're basically decomposing variation into node level traits and some shared smooth temporal trajectories. This substantially reduces the complexity of the network modeling by borrowing information across nodes or species and time to stabilize inference. This allows improved prediction and also identifies likely missing interactions. A last approach is one that's designed for count data. In this project, the goal is really to generate interpretable ecological insights from complex counts. The approach relies on a count matrix factorization using both binary and continuous latent variables. And the key contribution here is that we're able to really coherently relate latent factors to observed habitats. So we can cluster species by shared habitat preferences, and we can also identify regions of common profile. So this is providing a means of doing concurrent ordination and co-clustering of species and samples. And something that's really useful that comes out of this is also a method for identifying predictive or indicator species. So this provides a great illustration of how complex statistical models can lead to direct, actionable ecological insights. So stepping back a little bit, what these methods have in common is that they're all targeted at problems that arise from high dimensional, sparse data with a complex structure. And some common themes that we see through these methods is an emphasis on borrowing information across samples, exploiting latent low-rank structure in the data, and generally coming up with tools for scalability. And these methods have been accompanied by a lot of theoretical support. So while they've been motivated by the Lifeplan data, they've truly expanded the statistical toolkit in more broad ways. And so thank you, and thanks especially to the global team members who collected this data, and I'm happy to answer questions at the end, and obviously invite those who contributed to these methods, and David especially to join in if questions come up. Thanks.
Okay, thanks very much, Jennifer, for this excellent description of the statistical methods development in Lifeplan. So I will now continue by telling about our work on automated classification of birds, bats, mammals, insects, and fungi, and also say something about making ecological sense of the resulting data. And now, starting with the audio data, we have spent a lot of effort in developing improved bird and bat classifiers. We have, of course, not started from scratch, as there are excellent resources, such as BirdNet, which we have used here as a starting point. So what we have done is to fine-tune those existing classifiers to make them work even better with the kind of passive acoustic monitoring data that we have collected with audio mods, and tailoring the classifiers also to the specific locations from where we have collected the data. Now, a very important part of this work is the bird and bat sound annotation portal, PSG, which is illustrated on the left. So here we see as an example a spectrogram of a 10-second audio clip, which has been annotated by a bird expert who has not only listened the species that listed the species that vocalize in the clip, but also delineated bounding boxes around each vocalization. And now on the right-hand side, I have highlighted some of our key papers that describe how such annotations with a strong label can be used to improve machine learning-based bird and bat classifiers. And this is very much a continuous process, so I warmly invite all the bird and bat experts that we may have in the audience to take part to this collective annotation effort at the PSG portal. With image data, we have also built our classifiers on top of existing approaches, utilizing, for example, iNaturalist data and the megadetector and the SpeciesNet classifiers. This has enabled us to detect some 4.5 million animal bounding boxes from our global database of camera trap images. And we have then optimized our own classification models in an iterative loop of active learning, where we have generated a curated animal image library that we have then used to train, retrain, and validate our classifiers. Now here I'm showing one example of ecologically oriented analysis that you can do with this kind of audio and image data. Here at the northern latitudes, the availability of sunlight, of course, varies a lot over the season, and also along the latitude, so that in the north, during midsummer, it is day all the time. Now with Lifeplan data, we have asked how animals may adjust their activity under such conditions. In these data plots on the right, the x-axis is the month of the year, the y-axis is the hour of the day, and the curved lines, they show sunrise and sunset. And then all the small dots show the detections of these two bird species that I'm using here as an example. Now the song thrush on the left shows high activity around the lines, so it's a morning and evening singer. And then the willow warbler on the right appears to sing continuously during the day, with the exception that in the north it takes a break in the late evening, even if the daylight is there for 24 hours. The camera trap data are much sparser. Of course, we have much less detections, but also they allow for similar analysis of activity patterns, as shown here for the squirrel on the left that is active only during the day, and the fox on the right that is in the south active basically during the night only, but in the north where the night is very short or missing completely, it is active also during the day equally much as in the night. And now we had sufficient data to look at these kind of activity patterns for a bit less than 100 species, each shown here by a dot, and some illustrated by images as well. So what we see in this ordination plot is that most species really fit into one continuum that starts here from the day-active squirrel and ends to the night-active fox. But quite interestingly, some species fall outside this main gradient or main continuum, and hence their activity patterns differ from the rest of the species in more idiosyncratic ways. Here we have another study from the same data, where we have looked at the same Nordic data from the viewpoint of species interactions in bird vocalizations. So in this 1 minute spectrogram, the vocalizations of the willow warbler and the goldcrest are shown by the green and the red boxes. And these two species really seem to take turns to sing one after another. And now by analyzing the massive amount of recordings that we have statistically, we have confirmed that what we see here in this 1 minute segment is indeed the general case, so that these two species are behaviorally responding to each other. And furthermore, it's not only these two species, but many other species pairs as well follow similar turn taking. And this is especially the case for the small migratory songbirds with complex vocalization patterns, which by our analysis form the core group of turn taking. And this demonstration of turn taking among songbirds, I think it's a prime example of research that really requires the application of automated classifiers to massive amounts of audio data, as was needed to reach sufficient statistical power in this study. Now, while a lot of interesting analysis can be done using the Lifeplan data alone, even more can be done by combining different types of data. So in this example, we have combined systematic Lifeplan data with opportunistic citizen science data that was collected through a mobile phone application. And we combined these data types in a digital twinning approach that built on the complementary strengths of these different data types. And as I'm now trying to demonstrate using as an example of the lapping, this approach really made it possible to build real-time updating maps of observations at the very fine resolution, thanks to the millions of bird observations provided by the 300,000 citizens that took part to this project. So what we see here now is the predicted distribution of the lapwing during the breeding season. If I adjust for the migration, we see much lower probabilities because this species is right now coming back from migration back to the breeding grounds. Let's then move to the DNA metabarcoding data. And here you see how the data looks after the classification stage. So the upper table shows a small fraction of the main data table, where each row corresponds to one mallee strap sample. And then we have about 1 million columns that correspond to the species or the operational taxonomical units, OTUs that we found from the samples. And now one key feature of the Optimotu bioinformatics pipeline that we have developed is that it gives a complete taxonomic assignment for all OTUs, whether they are earlier known to science or not. And this is seen in the lower table, where there are many taxonomic identifiers that start with the word pseudo, meaning that we cannot reliably assign them to earlier known scientific names. Yet, this is very useful because it allows us to study the relationships between all OTUs, as for example, for some of them, we may estimate that they are likely to belong to the same genus, even if we cannot give the name for that genus. And one thing that we are especially interested about is the separation between those species that are previously described by science, here called the known taxa, of those that are earlier unknown to science, called here the novel taxa, and those species for which we cannot be sure if they are earlier known or not, called here the uncertain taxa. And as you see, on average, we can reliably assign maybe some 10% of all the animal and fungal occurrences in the Lifeplan data to earlier described species. Now for fungi, this proportion is essentially constant across the climatic zones, which is in really stark contrast with the insects, for which the proportion of unknowns is by far the highest in the tropical zone. So these data are still very fresh, but one of the very first questions that we want to ask about is what are these currently unknown species and in which ways they differ from the known species. And here we have been looking at this question separately for Europe and North America, which represent the more studied parts of the world with much prior ecological and taxonomic work, and then the other less studied parts of the world where we expected there to be a higher proportion of unknowns. And what we found is that in all regions there are more unknowns than knowns, as shown by on the right-hand side, the number of novel species per each known species being always higher than 1, and especially for the mallee-strapped insects in the less studied parts of the world. And what is more, our proportional estimation of number of unknown species is highest if we look at all the data, so the black bars and black dots, as compared to looking at subsets of data, which suggests that we may still underestimate the proportion of unknowns, especially for the fungi. And now what is really quite striking is that the spatial distributions of the unknown species are very different from those of the known species. So maybe not very surprisingly, the unknown species are much more rare in terms of how many observations we have on them. But even after controlling for the number of observations, the right-hand side graph shows that the unknown species have really much more restricted spatial ranges than the known species. So the y-axis in the right-hand side is logarithmic, so there is like a really, really big difference. Now, why this is very important is that current evaluations of the state of biodiversity, they are mostly based on the known species, which are just a minority of all the species. And as we know that species vulnerability to extinction is likely to be higher for the less abundant species and for the species with restricted ranges, this means that those current evaluations on the state of the biodiversity are unfortunately likely to be optimistic because the unknown species are likely to do even worse than the known species. Now, as Jennifer already described, much of our statistical developments, they related to joint species distribution modeling have been focused on computational scalability, as motivated by the very high dimensional Lifeplan DNA metabarcoding data. And I'm highlighting here especially 2 recent papers where we have made such scaling up possible through better utilization of high-performance computing resources, as well as the coral approach that Jennifer already described in more detail. So here is one example of joint species distribution modeling applied to cyclone sampler data. These are actually not the Lifeplan data, but the global spore sampling project data that we collected just before Lifeplan, with some of the teams continuing also with the Lifeplan project. And what is especially interesting with this kind of analysis is to look at the taxonomic or phylogenetic signal, so asking whether related species show especially similar environmental responses, as could be expected based on their shared evolutionary history. Here we found a high phylogenetic signal, especially in terms of the seasonal sensitivity of fungal sporulation, and a somewhat more moderate phylogenetic signal in terms of the climatic sensitivities of the species. Now, many of the Lifeplan cyclone samples, they contain an insect, a spider, or some other kind of a bug. And this is, of course, especially the case for the summertime samples, as is shown by the left-hand most panel in this figure. So we realized that this is really an opportunity to ask about the role of insects in fungal dispersal by comparing the samples with and without insects. And now, as shown in the middle panel, the samples with insects or any other kind of a bug, they have much higher fungal richness than those without the insect. And now, as shown in the right-hand panel, this difference remains also after controlling for the confounding factors, most importantly seasonality that influences both the insects and the fungi. So Diptera appear to carry an especially high diversity of fungi, whereas Hymenoptera and spiders, the Aranea, they appear not to influence fungal diversity almost at all. Now, a joint species distribution model applied to this data suggests that not all fungal groups are equally dispersed by insects. So here the red color indicates those fungal species that are more likely to be present in samples that have an insect, shown separately for latitude 0 and latitude 60. And what we see here is that especially in the Basidium mycota, there are groups of closely related species that all seem to benefit from insect dispersal, or at least they are present in samples with insects. So we think that these global data are really useful in understanding the role of insects in fungal dispersal, including the matchmaking of what kind of insects carry what kinds of fungi. All right, in the interest of time, I couldn't go through more examples in my presentation, but I'm very happy to hand next this over to Brian Fisher, who will tell specifically about our research in Madagascar.
Hello, everyone, and thank you, Otso. And this next talk really builds upon everything you've seen so far, including the biostatistical methods developed, but it also builds on an incredible effort by the team in Madagascar, where we had over 100 staff working tirelessly to keep the equipment going. For those of you on the call who ran a local spot in the tropics, you have my utmost respect, because you thought you got a job to do bioscience, but it turned out you were doing biomanufacturing daily to keep the equipment up and running. Just to give you a prelude on today, we're going to focus just on Madagascar. And there are so many things we could be talking about. For example, how in the first year of data collection, we learned more about the arthropods diversity than we had in the last 200 years. But we're not going to talk about that. We're going to focus on, I think, a really important topic that's directly related to conservation. And that is For decades, we built models based on vertebrates and plants to understand biodiversity patterns within Madagascar. Well, today, thanks to Lifeplan, we can finally test those assumptions. Now, why Madagascar? Madagascar is special for two reasons. One, it's had a long history of isolation, and it's geographically placed in the tropics. So because of that isolation, it broke away from Africa about 120 million years ago, along with India. India then moved north, dropping the Seychelles, hitting Asia, forming the Himalayas. Madagascar has been isolated for about 80 million years. And one true global principle is that isolation results in high endemism. And in the tropics, we have high diversity. But the question is, within Madagascar, what's driving those patterns of diversity for those endemic taxa? And it's been an industry of activity to develop models that are tested within a certain clade, like vertebrates, reptiles, or lemurs even, that try to explain the patterns within Madagascar. And Lifeplan data provides a perfect way to actually test if those models that were developed, those drivers for patterns of endemism within Madagascar, are applicable or are the same across taxa. So that was the problem we could use our data to solve, which is a very important problem for conservation. And let's look at our approach in Lifeplan. As you know, we have the standard methods applied to Madagascar very intensely. We have the arthropods, which we can use CO1 to look at diversity patterns. We have the cyclone sampler for fungi. We have audio for birds, and we have the camera for vertebrates. But I want to convince you that our sampling was very precisely designed to capture the full diversity of the island. So on the far upper left there, you can see the ecoregions and our distribution points. They encompass all the major ecoregions, which include vegetation. In the center there, we have a map of the climate zones of Madagascar. And our samples include all the climate zones of Madagascar. And on the far right, one important point is all our samples were located in the best forest we could find. Unlike the rest of Lifeplan, we really reduced the impact of humans on our sampling. We tested for this, but we actually wanted to make sure we were capturing the best possible forest. And like in other sites on the lower graph, you'll see that our coverage is not always complete for a single site because of mostly damaged equipment, and while we waited for replacement screens from China, a new microphone, or fixing the motor in the cyclone sampler. So this is just a summary of the coverage of the data, and I think it's convincing that we have enough data throughout with the standardized sampling that we can actually then test this. So we have same sites at the same time in the same protocols, and that allows us to overcome a lot of the restrictions of doing this study on various data sets. So before we get into looking at drivers, there are two really interesting patterns I want to talk about. And this here we show each of the main data sets in a column. So we can look at arthropods as the first column. And if it's red, it means there's more of that. And the first upper left graph, we have species richness. You'll see that in that graph, looking at arthropods, the richness is in the northeast of Madagascar. That's also where it's wetter and warmer in Madagascar. And we can also then look at two levels of endemism. And this endemism is important. The one below it here shows if the species is found only in Madagascar, or the OTU, only in Madagascar and not elsewhere, it's labeled as endemic. However, we also wanted to make sure that we weren't biasing this by the maybe low coverage of sampling, like in Africa, for example. We actually then looked at another level of endemism we call locally endemic, and that is we're looking at within samples that are found only locally within Madagascar, and that patterns are quite similar. So arthropods, the northeast is the rich, endemic taxa are found throughout the island. Locally endemic are also mostly in the northeast of Madagascar. And that is in contrast to, for example, fungi. Fungi have about the same local diversity, but the patterns of richness and endemism are quite different. And if you look at birds and mammals, it's mixed. Some of the patterns are similar to arthropods, but many are not. So the end result is the hotspots of diversity are not the same for each of those main kingdoms that we're discussing here. Now, we have a unique chance to also tease apart and reanalyze the data by just looking at endemic taxa compared to non-endemic taxa. And I think this is really the strength of this study in Madagascar. And if you look at arthropods, which is the CO1 data, first columns in the bar graphs and also in the line chart, versus the fungi, which is next, followed by the audio and then the images, you'll see that for all the arthropods or other, and the fungi, let's do that comparison, there's about an equal number of endemics and non-endemics for those taxa. But it's only the arthropods that have a high singleton rate and have really true microendemism across the island. So the end result is that arthropods see the world on a much finer scale, and that the endemism is actually because they have restricted taxa that have a small range. And the lower right graph shows that controlling for detection rate at 3 levels, that the pink versus the light teal color, you'll see always that the ranges for the non-endemics at any detection rate is larger than for arthropods, than for the endemic taxa. So we have 2 generalized patterns that arthropods show hotspots, different pattern of hotspots, but also a higher level of microendemism. So now we want to ask the question, what's driving these surprising contrast? So we can look at the question broadly and say, is it geography that's driving these, or is it the environment? That would be like the climate, for example. And then we used linear models to model all these variables across the 53 sites. And there was, I think, a really exciting discovery here that the answer is kingdom dependent. So for arthropods, geography drives the difference, while for fungi, environment drives the difference. And you can say, wait a minute, isn't that the same thing, the environment versus geography? What this is saying is that for arthropods, even if you have the same habitat, the same climate class, you can't predict what species you'll find there if it's distant. In other words, geography plays a major role. While for fungi, if you can actually know the class of the environment, you can predict what species will be found there. And for birds and mammals, it's a mixed signal. At times, one is stronger than the other. This is for all the taxa. And the next is looking at just the endemic and comparing that to the non-endemic. And this is where I think is more interesting from Madagascar, where we want to look at and apply it to conservation for the endemic lineages for arthropods or fungi or birds and mammals. But the pattern holds up that for arthropods, geographic distance dominates for those. So I think I keep having time here. I can keep going to the last, I think, important point here is, so what does this mean? So what? Well, in Madagascar, in fact, next week, there's a major meeting that's occurring that's deciding, prioritizing what are the next protected areas that should be added to the protected area system of Madagascar. And these results have direct relevance for that. Because what you would decide for example, birds and mammals or fungi is not what you would decide for arthropods. For arthropods, geography isolated sites potentially harbor species that are not yet captured by the current protected system. So that it would be better to make sure that there's a distributed, even if they were smaller parks, to capture diversity rather than a few large parks that would maybe have larger populations of lemurs, for example. You would still be missing important arthropod lineages if you only had a few large spots versus many distributed sites across Madagascar. And those differences are quite different. So in the end, to conclude, the take-home message is that thanks to Lifeplan data, we could actually develop a first ever test of whether or not these within Madagascar drivers of diversity are applicable across kingdoms. And the answer is no, that you have to have specific models for each kingdom. But it also calls that arthropods do need to be included in conservation planning because their model captures the most diversity and actually is quite distinct from other models that would be applied for fungi, vertebrates, or birds. Once again, thank you for the teams that have made this possible. It's not an easy task, and look forward to reading your questions in the chat. Next up, we'll hear from Tomas. Thank you.
Thank you, Brian. Okay, you can see my slides. Good. So from Madagascar, we will move back to the global level, and we will think about what major patterns and processes we have discovered and understood across the world. And the important thing before going wild regarding global inference is clearly to think about what the samples represent. So, I mean, we're talking about abundances, we're talking about densities, we're talking about species richness, but we're still basing that on samples. And as a very concrete example, we can think about a malaise trap. So here's a malaise trap, and our samples end up in the little bottle at the top. And the first obvious question to ask is, can we even characterize who reaches the bottle? What's in the bottle? Can we trust the description of that as based on our high throughput methods? And that we have tested very carefully. We have done so by taking full malaise samples like the one on the left, then metabarcoding all of that stuff, and then picking out each and every individual from that sample. So we know who was there as a physical individual. And then we ask, okay, so if we know the number of individuals of a given taxon, in this case a bin, in the sample, can we predict the presence of this? Will it be found in the metabarcoding soup? And we find, yes, indeed, even for very low numbers of individuals, we will find it in the metabarcoded data from the soup. But we can also ask the other way around. So if we see a few reads from the metabarcoded data, how likely is it that the same taxon actually occurs as an individual? And here we find that for low read counts, in quite a few cases, there is no individual to be found. So we're fairly optimistic about this. It means we can detect who's there, but we can also detect a lot of other taxa, which are likely stuff that came in the gut contents or in the surface of these arthropods and got sequenced along with the real individuals. So we get an even larger sample. So we get a check on this. But the other question, which I think is way more interesting and way less explored, is saying, okay, but for us to detect a bug in that bottle, it needs to end up in that bottle. Now, how does the number of bugs ending up in the bottle actually relate to the reference community? Because we're not inferring stuff about numbers of individuals in a plastic bottle. We want to say something about global variation in arthropod communities and densities. So how do we translate from what we see in the bottle to the true world? And here we've done a lot of kind of calibration studies with the idea that if you mark individuals in the reference community and you blend them with the rest of the wild individuals, and then you look at who is later caught in a trap, then you can actually infer the diffusion rates of these, how they move through the landscape, their mortality rates, and their capture rates. And from that, you can actually back calculate. So if we see so and so many individuals in the bottle, how many other individuals will be lurking around in the surrounding forest? And the answer is quite a few. So these are conversion factors for different insect groups, and they are in thousands of individuals. So for every individual that you find in a malaise trap of small wasps, there will be some 50,000 for every individual. And this varies across groups. Some are easier to observe and some are harder to observe. But as a rough rule of thumb, we can say that if you multiply the number of individuals that you see in the sample by 30,000, you will get an estimate of the number of individuals in the surrounding hectare. What's also important is that these conversion factors actually vary around that 30,000, which means that you shouldn't take the number of individuals per taxa in your bottle as an immediate representation of the relative abundances of these species in the surrounding, because some are easier to trap and others are harder to trap. So if we see in this bar plot on the left, this taxonomic division of the sample, we should actually infer that the proportions of these taxa in the reference community are slightly different because of this difference in observability. And as a rough number, we can also, based on how many individuals we tend to trap and what these conversion numbers are, we can infer that on average, there may be something like 4 million insect individuals per hectare in a boreal forest. And if you were worried about catching each and every one of them by your malaise trap and making a major environmental crime, you can now relax, because what this shows is that on average for every individual you catch, 30,000 will go free and live a happy life. Good. We turn from malaise traps to sound. Together we published this exploration of global soundscapes last year. And here, as Otso was describing, because global ecologists are doing so well on identifying birds, here we can actually convert all these recordings to fairly reliable records of what birds were present in the surroundings, and we can describe rhythms in numbers of bird species vocalizing for different hours of the day and different days of the year. We can compare bird species numbers from these recordings between natural and urban green spaces, and we can even look at differences in identity, saying there may be a few more bird species in urban green spaces, but they are different than the ones we see in the forest. Okay, this is very cool, but then we have a lot of sounds which we cannot make sense of because we don't have as many bat watchers as we have bird watchers across the globe. And the question is, we have 2 centuries of recordings of bats, but we don't have the same kind of global identification facility. So what do we do? And here, one of the most recent innovations is to say, okay, we can't do the traditional thing where we train a model using a reference library where it learns the individual species from there. But how about using simply the patterns present in these vocalizations? And this is Panu Somervuo's idea. So he has sampled Lifeplan ultrasound data from high frequencies, and then he has used a neural network to kind of characterize these recordings in very many different dimensions. So 1,500 different dimensions. And then you can, across these dimensions, calculate the distance between the different clips, cluster them up, and now you have kind of operational or audio taxa or operational audio units, which you can, so you have brought structure into chaos, and you can go through these individual clusters and ask what they really are, and you can refine it by clustering by finer criteria or laxer criteria, and thereby characterize the unknown soundscape. And these pictures are just representations of individual clusters, where the first row are clips which are close to the center of the cluster, and then if you go down, they are further and further away, and you can see by eye that they are fairly similar within clusters. Okay, then we have looked at insect biomass. We already had a peek view of the data. We have been trapping insects for weeks across all of these different sites. We have weighed them, and now we can ask, what are the rhythms of the cycles of in insect biomass across different parts of the world? And the simplest, I hope you can see this fine animation. The simplest thing to do is to simply fit this to periodic functions to allow interactions between the periodic functions and latitude, so that season affects the biomass rhythm a little different in different places, and now you have a global model of insect biomass. This is very descriptive, but you can also ask things like, when taking into account what season it is and where you are, does insect biomass vary with the human impact, with the human footprint? And here the finding is actually that at low latitudes, the effect is very little, but when you go far towards the poles, then you have a positive impact, which isn't hard to imagine, in fact, because these are very low productive areas. And I guess one thing that we as humans do is that we add nutrients and energy. So this is a very cool finding. Then Jennifer already gave a sneak peek of this result. I still want to return to it because it's so very cool. Because from an ecologist's point of view, the idea here is that, think about all these biodiversity indices that we have invented, from evenness to Simpson and Shannon and you name it. What they all describe are two features of biodiversity: how many species you have and how evenly they are distributed across taxa. What that means is that they can all be inferred from the taxon accumulation rate, which depends on the species abundance curve. Now, if we can learn the shape of that curve, and if we can make it covariate dependent, so we can look at how steep it is depending on the environment, then we have a strong predictive tool for every part of global biodiversity. So by fitting these lines, you can estimate all of these and very many more biodiversity indices, and you can create nice animations again. And you can also go for scenario analysis because you can ask, okay, so if now biodiversity in this wide sense depends on the human footprint index and on the climate, like actual evapotranspiration, and we know the shape of that dependency, then what happens if there were no humans? Oops, we would have 60% more species in a sample of a given size. Or if we increased evapotranspiration by fiddling with the climate, we will have a substantial increase of up to some 10% in species richness in a sample of a given size, which is very cool. The final point, and I have no idea how I'm doing on time, but likely poorly. The final point that I want to make is that Lifeplan data, they are not generally on interactions. You can infer co-occurrence, and then you can indirectly think, oh, maybe these species affect each other. But there are also direct observations of interactions. Because for the insect data, well, Otso was already talking about the fungi and the insects ending up in the same traps. For the insects, we have a lot of data because each individual is the host of a lot of different microbes. We can sequence those both at the level of individual insects and at the level of a malaise bottle with a known community composition, and we can already here see that there are very cool patterns to find in terms of here, for instance, symbiotic taxa like Wolbachia, which happen to amplify with the same markers that we use for the insects themselves. So what I wanted to say in slightly too many minutes is that we're fairly confident that we can actually characterize the sample contents by these high throughput standardized methods. We are making progress on actually converting what we find in the sample to what is happening in the true inference community. We are thinking of ways of automatically identifying taxa for which there are no kind of comprehensive reference libraries as based on patterns in the data and efficient annotation of the clusters that we find. We can predict biomass and biodiversity from environmental covariates and human disturbance. And that is very nice because if you have a predictive model, you can also imagine things and do scenario analysis. And the next thing, which we haven't really started, but where we will have the data, are these species interactions, which are still hidden in the bottles. That's it. Thank you.
Okay, thank you very much, Tomas, and also thanks for all the speakers keeping very well their time. We are 1 minute late from the original program, so that's exceptional. So let's then go back to the bigger picture and the conclusions, some concluding remarks, and then questions and answers. And I have 2 slides about the conclusions. So first, at the very general level, what have we learned by running the Lifeplan project. And I think the first thing that we learned is that systematic and comprehensive global scale biodiversity research is indeed possible. We are extremely happy about the data set that we have jointly generated over the past 6 years in this project. But as you have heard in the presentations, that has required a lot of resources in terms of researcher time and funding, and especially the effort of the hundreds of researchers who have contributed their valuable time to the globally distributed sampling. Without that, none of this would have been possible. So I think one key take-home message for future projects is to think how to streamline the sampling even more, to make it even more cost efficient and even less resource demanding, especially in terms of the researcher time. And my second conclusion here is that biodiversity research is definitely taking huge steps towards automated sampling, classification, and inference. And we have been very happy with the Lifeplan project to be part of these global developments that many research groups also outside Lifeplan have been contributing in the recent years. And this is indeed badly needed, as most biodiversity is still unknown and we are losing it at increasing rate. So we feel that these kind of methods that we have presented here, they are more or less the only way to survey biodiversity in a sufficiently efficient and fast way. Okay, then my second slide is about the future. What are the next steps after Lifeplan? And we have basically spent 5 to 6 years mainly to generate the data that we have shown here. As we have shown, we have done some very interesting analyses, but I think we have really scratched the surface for the reason that the data has been available for us only for a very short time. A year ago, basically, we had none of this data available for our own analyses, and more data have been becoming available over the past months, weeks, even days. So now we are at the stage where we have all the data available almost, but the project is ending. And I think this is a very typical situation for many research projects. We will most likely need at least another 6 years to make the most out of this data, and those of us who have the possibility to continue working with this data will for sure be very happy to spend those next 6 years to make the most out of it. And now, along with these scientific papers to come, as Bess already described, we plan to publish the raw data as specific data papers to enable the global researcher community to take the full advantage of these resources and make most out of these comprehensive biodiversity data. So thank you very much for staying in this webinar. We hope that you found this interesting. And next we will have a questions and answers session, and some of the questions I see have already been answered in the chat, but maybe some of them require some would benefit from a discussion. Yeah, there were some earlier comments about whether we will make the slides available or whether this recording is available, and Bess has put there a link that the recording will indeed be in the YouTube channel, but unfortunately we cannot share the slides as such because they have a lot of unpublished and copyright material. Would any of the other panelists like to highlight some of the questions that have been asked?
There was the question about the turn-taking versus spectral overlap, which I thought was very interesting, and you provided a very short answer. You said yes.
Yeah, the question was whether the turn taking was found especially for species that overlap in their vocalization frequency, if I understood the question correctly. And yes, we found that this is the case because those species that followed turn taking, they were all these songbirds that have like a very broad spectral bandwidth that they sing at a broad range of frequencies and overlapping frequencies.
There was also if that topic is covered, there was also a great question about the biodiversity data as an opportunity for more interaction modeling. So the question was whether the team sees potential for combining these data sets with the concept of metawebs, the regional pool of potential species interactions, to explore how local networks assemble across environmental gradients. And I think that the answer is a resounding yes. That's something that we've been working on and is really interesting. And the question highlights that there are a couple different concepts of networks that are useful to keep in mind and to think about how they interact. There's the sort of larger fundamental network of potential interactions. And then there are the realized networks at any given point in time and space, which may vary as well. And so modeling those realized interactions across time and space is one approach, but also those should be informed by the meta network, right? That we shouldn't have interactions be possible at any given point if we know that they're not in the meta network. So there are a couple different threads. One involves sort of link completion in the meta network, and then another in using the meta network to improve analysis of dynamic and spatially varying networks. Both very interesting.
Thanks, Jennifer. I see that there's a question by Errol about whether the Lifeplan network could be leveraged to generate some practitioner-led publications, and especially those who have been active in implementing the actual research in the field. And I think this is a very good question. This is something that I think we could discuss tomorrow when we have kind of another meeting specifically with the sampling teams. So we could bring this up there and then discuss how they feel about this suggestion. Of course, like from our point, it sounds like a great suggestion, but I think it's very much up to the teams, sampling teams, that how they feel about this. But thank you for the suggestion.
And I just put something in chat. It's a paper by Jennifer, and it's not on Lifeplandata, but it shows the great value of Lifeplan, because that's something dealing with, network data from some remote part of Greenland. But unless we had been brought together by the Lifeplan project with this exact synergy objective, I would never have met Jennifer, and that thing would never have happened, which goes to show just how much fun we have had and how useful it has been.
And I could address one last question in the chat, which is, will the trained models that we presented be published for transfer learning? So we will be publishing everything. It's a part of our funding agreement that we have to publish everything open access. The only things I think we can't publish openly is all the raw audio and image data, because there might be people in there. But aside from that, we do publish everything open access, and certainly the models.
And then there was a question about whether we are considering some region-specific manuscripts. Now, of course, much of our primary interest is in the global patterns and then patterns in Madagascar, because our data are especially rich and unique about these aspects. But as I already illustrated by some specific analysis on the Nordic bird and mammal communities, of course, we are also interested in doing like a region-specific analysis whenever there is some inspiring question for which region-specific Lifeplan data can be used for. But then we especially encourage the sampling teams to make the most out of their own data, and we are unfortunately a bit late from our original plans in delivering the data back to the sampling teams, simply for the reason that developing like sufficiently reliable classifiers has been quite challenging, and because we haven't had like robust enough species lists, what species were found from each sample available, we haven't been able to deliver the data back to the teams as fast as we hoped for, and we are working very hard to make this happen as soon as possible. And then we of course hope that this will inspire a lot of region-specific manuscripts that are either led by the sampling teams or by the Lifeplan researcher team, whichever is the most natural solution for each case.
And Renata is offering annotated bat data from Natal. Works for us. We will ask Kata to get in touch with you.
I see that Lucas is asking a question that whether these vocal turn-taking patterns were identified from manually annotated data or from the classifier results. And they were indeed inferred from the classifier results, because that was the only way to make the most out of all of these hundreds of thousands of minutes of data that we had. It was not possible to annotate them manually, but then Lucas is asking the very good question that if these and as these classifier results were used, how did we account for potential biases in detection probability? And we manually annotated a subset of the data to validate our classification models for all of these species. And that actually led us to exclude some of the species from the analysis because we realized that our classifiers were not reliable enough for those species. But then the question continues especially about these biases in detection probability caused by overlapping vocalizations. And this is indeed a very good question, that when we have multiple vocalizations at the same time, then we have a harder job in classifying the species because there is more noise around the vocalization of each individual species. And because of that reason, we may, just as a statistical artifact, estimate that the species do not like to vocalize at the same time. And actually, this is a pattern that we detected basically for all pairs of species, that there was kind of apparent avoidance of vocalization at the same time. And now that, I think, is partially a statistical artifact, but then this turn-taking, that they like to vocalize 4 to 6 seconds after each other, that is not like, that's very hard to believe that would be any statistical artifact. All right, hey, I think we are a little bit over the initial time. Looks like we are maybe out of questions, but at least we are out of good answers to any questions. So maybe this is a good time to thank everybody again for joining this webinar. If there are some burning questions that remained unanswered, feel free to email us, and then we can continue the discussion afterwards.
Yes, Cremena has been trying to ask in 2 channels and she can't transfer, and I think those questions go to Brian. So if you could email Brian, that would be great.
Yeah, feel free to email me. And in terms of our anthropogenic study, we used just human HII, and it was very modest. Our sites were in parks. That explains why they were modest. And it was not a major, in the glimmer analysis, it was not shown to be a substantial impact. So it did not structure biodiversity compared to like climate, for example, or environment or distance.
Okay, thanks, Brian, and thanks all the positive feedback that I see is coming there. So it's time to close. Bye.